Hello, everyone. Good morning, good afternoon, and good evening. This is Miss Kate, and for this lecture video, we will be discussing the continuation for Enterobacteriaceae. Apologies for my voice. The flu caught me. Anyway, the show must go on. So let's start with E. coli. So these microorganism or bacterium to be specific is also a gram-negative rod-shaped bacterium. If you have remembered my previous lecture, this morphology of bacterium or bacteria are the form that you'll often encounter in both clinical and laboratory settings. Now, these bacteria, this Escherichia coli, are commonly isolated from the colon flora. So that is why most of the books refer this bacterium as the colon bacillus. So meaning when we say colon bacillus or commonly isolated from the colon flora or colon microbiota, this would only means that they are a regular part of the microbial community that resides in the large intestine. That is why they're also referred and considered as commensal microorganisms. But what does this term mean, or what commensal means? I know I have already discussed this one, but in the context, just for review, a commensal bacterium is the one that lives in a host without causing harm. Instead, they coexist peacefully within the body. So, in this type of bacterium for E. coli, they coexist peacefully within the body, particularly in the lower intestine. So, how these microorganisms or bacteria actually helped us? So, these bacteria have actually adapted to thrive in the nutrient rich environment of the gut. So, meaning, Escherichia coli contributes to the host health by aiding in digestion or absorbing vitamin K and vitamin B, and also competing with the potential pathogens. But then again, class, please take note, while they are generally harmless, under certain circumstances, like there's a breach in the intestinal wall, if the patient has undergone surgery, or a weakened immune system, so if the patient is suffering from cancer, autoimmune disease, or a patient is pregnant, so these organisms or bacteria can become opportunistic pathogens, meaning they actually grab the opportunity while the host is immunocompromised to cause infection. So when you read the updated journals, please take note that Escherichia coli is now classified into two. We have the opportunistic, these are classified under opportunistic and as a primary or true infection. So it is why are we telling, why we explaining or including this concept? It is essential for us to recognize that there is actually a dual nature of these bacteria, and they could be vital members of the gut microbiota of our intestines, supporting overall health. But then again, we need to remember that they have also the potential cause to give infection if they are translocated to other parts of the body. I think I have already discussed this one. So, if the colon bacillus or if your Escherichia coli thrives in your colon, normally in the colon, if they are translocated to other parts of the body, say for example, in your urinary tract, E. coli would cause UTI and so on. So, we need to remember this one. So, what we have here is the picture of a microscopic examination of your Escherichia coli. As you can see here, it's a gram-negative rod-shaped bacteria. So usually when we describe this in the laboratory, it is referred to as coccobacilli. So it is a mediator of your cocci and your bacilli. But then again, we could um, differentiate and compare this with our gram-positive Staphylococcus aureus. The Staphylococcus aureus is the complete opposite of our Escherichia coli. As you can see right here, Staphylococcus aureus is cocci, and it shows a gram-positive reaction. As we could see here, on the other hand, bacteria with a gram-negative reaction. Next, okay. So, your Escherichia coli has an amazing amount of DNA being swapped about among the enterics. If you could remember, I always labeled Escherichia coli as the mutant one. So it is a very versatile bacterium that usually plays a significant role both as a commensal and a pathogen. And it has the capacity to receive virulence factors from all of its neighboring bacteria, and that includes Shigella and Vibrio, if you could remember. So there its disease spectrum is so wide. So it includes gastrointestinal infections, and that includes diarrheal diseases right here, and it could also includes UTI and some central nervous infections. So please remember that again, E. coli is now recognized as a crucial human pathogen. Okay? It is an important human pathogen that is associated with diverse clinical syndromes, from UTI to diarrheal syndromes, or from extraintestinal syndromes to CNS infections. So aside from E. coli as an opportunistic and as a true pathogen, E. coli serves as the primary marker of fecal contamination in water quality testing. Again, your E. coli serves as the primary marker of fecal contamination in water quality testing. Why? This is due to several reasons. Number one, your E. coli again is present in the gut, right? So E. coli is naturally found in intestines, and its presence in the water indicates that there is actually a contamination with fecal matter. And if there's contamination with fecal matter, it may also harbor other pathogenic microorganisms. So other than that, E. coli is an ideal indicator for fecal contamination because number one, again, it is easy to detect, and usually it is non-pathogenic in its commensal form, and its survival skills or its survival characteristics in the water are similar to those of those any enteric pathogens. That is why E. coli is known to be the most versatile bacterium. So aside from it has a dual role, like a commensal and then a pathogenic or a true pathogen, it also serves as a primary marker for water quality testing. So right here is an example of your favorite beach, your Boracay. So how you know that the enterics are transmitted by the fecal oral route, right? So how will you prove that the water is actually fecal contaminated? So again, remember that your E. coli is a primary marker for water quality testing. So you see, E. coli is a coliform, which means that it is a normal inhabitant of the intestinal tract. So again, think of E. coli, coliform meaning it inhabits your colon. So E. coli is normally not found outside the intestine. So if you find E. coli in the water, it does not necessarily mean that E. coli is causing the diarrhea, but it does tell you that there is fecal matter in the river or in the beach, and that some enteric may be responsible. So that is why it actually happens to Boracay. So a few years ago, Boracay faced a serious problem. So hidden beneath its beauty, this beautiful beach. So in 2018, if I'm not mistaken, pre-pandemic, the island was temporarily shut down for 6 months. Why? Because it was being overwhelmed, particularly from a type of bacteria called fecal coliforms. So again, what are these bacteria? So these are the bacteria which are commonly found in humans and animal waste. So it's not just humans. So they were actually discovered at dangerously high levels in Boracay's waters. So the situation was so severe that the island's once beautiful, pristine beaches were actually at risk of becoming a public health hazard. So that is our job to minimize these risks. So again, Escherichia coli can indicate the presence of these pathogens that causes these diseases. So when you have high levels of these bacteria in the water, just like what happened in Boracay, it means that there is likely a sewage or other waste contamination, making the water unsafe for swimming or other recreational activities. That is the importance of your E. coli. So, for species of colonizing humans, infection may result when a patient's own bacterial strain or endogenous strains establish infection in a normal sterile body site, and that happens to your E. coli. So, how, what are the virulence factors? So, what are the things that could, or the factors that could actually help E. coli establish infection? So number one, we have here for movement and other some surface features. So most of these bacteria of E. coli can move around. So using the tiny whip-like structures called flagella. So they have also sticky hair-like structures. So those small hair-like structures, and that is known as the fimbriae. So those are the fibrae. So the fibrae help them attach to the surfaces. They have also special structures. Right here, we have here the sex pili. The sex pili allow them to share genetic material from with the other bacteria. So if you can remember, through the sex pili, it serves as the bridge that would allow these genetic makeups from other bacteria to be received from the recipient bacteria. So we have also here specific markers on the surface. We have here O antigen, the H antigen, and the K antigen, which we have already discussed a while ago. And we have also the sugar fermentation. So E. coli can break down several types of sugar, and that includes glucose, lactose, trehalose, xylose to get energy. Aside from that, we have here indole production. So they can actually convert a substance right here called tryptophan into another substance. And that is what we call as the indole. So we could actually detect this indole formation or the product indole in a simple test. So through indole test. We have also here acid production. For acid production, when they break down glucose, if you could remember, they produce a lot of acid, right? So this can be shown with a test called methylene red test. So methylene red test, which turns red if the bacteria produce a lot of acid. We have here Vogue's per scour. So E. coli do not produce a chemical called acetoin. So kindly write it down, acetoin. That is ACETOIN. So when they break down glucose, so acetoin is not formed in E. coli. But if your Vogue's per scour is positive, that bacteria could actually produce acetoin when they break down glucose. But that is not the case for E. coli. So your acetoin production is tested using the Vogue's per scour test. So we have also here other enzyme activity, and that includes your hydrogen sulfide. So your E. coli does not produce hydrogen sulfide. It does not produce your enzyme DNase, which breaks down your DNA. They do not produce urease, an enzyme that breaks down urea into what is the byproduct of urease to urea, breaks down urea to clinical chemistry, that is ammonia. They do not have an enzyme called phenylalanine deaminase as well. So for the citrate, they cannot use a substance called citrate as their only source of food and energy. So Excuse me. As you can see here, I have here, excuse me, this is what we call as the Imvik reaction. Vogue sour and your citrate. For your E. coli, the citrate, sorry, the Imvik reaction is positive, negative, negative. Again, the Imvik reaction for E. coli is positive, positive, negative, negative. That only means that for in for tryptophan, and that is positive. For methyl red, that would be positive as well. V for your citrate, for letter C. Okay, so for next week, we will be actually discussing of the biochemical characteristics in the laboratory, and you will be performing some of them as well. So let's proceed. So right here, we have the different agar. So what you can see here in front of you, an example of your Macaque agar. So if you could remember, your E. coli is a rapid lactose fermenter. So you can see here in the Macaque agar, it shows a lactose positive colony. So meaning it is a lactose fermenter with a, usually here in picture A, you can see precipitated bile salts. So this one. So remember that you have your bile salts with your Macaque. Usually it is precipitated. So again, just a review, do not forget that your Macaque is a selective and differential medium. Okay, it is both selective and differential medium. It is selective. Why? Because it has bile salts and crystal violet that inhibits the gram positive bacteria. It is differential. Why? Because it could differentiate the bacteria enterics to be specific from the lactose fermenter and from the non-lactose fermenter. Do not forget as well the pH indicator that could actually tell us that if if fermentation takes place. So we have here the neutral red. So neutral red is the pH indicator. So again, why are these? We have these lactose fermenting colonies. So we could tell that E. coli can ferment lactose, which produces acids, right? So lactose produces acids, which lowers the pH. Again, if there's fermentation, so for E. coli, the E. coli rapidly ferments the lactose, produces acids, which lowers the pH and causing the neutral red indicator to turn pink. Therefore, lactose fermenting bacteria form pink colonies. So pink colonies. What we have here is another agar, and that is your EMB agar. It has a beautiful green metallic sheen right here. So just like your MacConkey agar, your EMB agar, which stands for eosin methylene blue agar, is again both selective and differential. Again, eosin methylene blue agar is both selective and differential, meaning it is used primarily isolating for fecal coliforms. So again, your EMP is the primary agar used for isolating fecal coliforms, your EMB. Why is it selective? So we have also some ingredients. So we have the aosin Y, and methylene blue. We have the aosin Y, just put Y here, and then the methylene blue. So your ASNY and your methylene blue has the same purpose and has the same function of your bile salts and crystal violet of your Macaque. So therefore, your ASNY and methylene blue also inhibits the growth of your gram positive, promoting the growth of your gram negative. So that's selective. We have also here some differential ingredients. So other differential ingredients that includes your lactose and also your ASNY and your methylene blue. It actually helps as well for the differential ingredients. And your your methylene blue also acts as the pH indicator. So just like your neutral red. So your neutral red and your methylene blue. So again, we have here a green metallic sheen. Green metallic sheen. So therefore, Escherichia coli, just like the other for lactose fermenter, produces strong acid production. So again, in your E. coli right here, it produced dark colonies with a distinctive green metallic sheen. Why do we have a green metallic sheen? This is because due to the reaction of the between dyes. So the dyes that we are actually using is the methylene blue. Methylene blue has a very strong color, and with a combination of the strong acid production. So for blue, and we have also a strong acid production. So we have here a beautiful greenish metallic sheen. These are usually seen in Escherichia coli. So next. So we have here the differentiation between your lactose fermenter and your non-lactose fermenter. So again, your McConkey agar right here. So it contains your selective ingredients and the differential ingredients. We have here the bile salts and the gristle violet, which inhibits again the positive bacteria, promoting the growth of the gram-negative bacteria. We have also the lactone, which would allow us to differentiate bacteria from non-lactose fermenter and lactose fermenter. Of course, we can't tell there is we don't have and that is why we have here the neutral red, the pH, therefore lactose fermenters show, while your non-lactose fermenters colorless colonies when inoculated in your McConkey agar. So we have here some virulence factors. So these virulence factors is very important when we study the diseases of each bacterium. So virulence factors is what we have here is mucosa or the mucosal interaction. So we have the pili, specifically the pili, but what was not included here is also the fimbriae. Fimbriae also initiates mucosal adherence. Mucosal adherence. So this is letter L, by the way. This one's letter A, not an exclamation. So, what is the pathogenesis of the mucosal interaction? So, if you just think of your mucosa as inner lining of your intestines. So, we have the bacteria that has tiny hair-like structures, if you could remember, and that is your pili and your fimbriae. So these bacteria has this tiny hair-like structures. So these bacteria actually grab onto and stick to the mucosal lining of your intestines. So this factor actually helps them stay in place and start colonizing. When we say colonizing, that means it refers to growing and multiplying in bacteria. So that is the importance of your pili and fimbriae from adhesion. So they stick to the mucosal lining of your intestine. So no matter how you trying to or how no matter how our body tries to wash them out, then they cannot wash these bacteria out because of these virulence factors. I think I have already discussed the types of diarrhea, so I should not go on and discuss anymore to repeat it again. No, so you just have to correlate the types of diarrhea and the virulence factors, the general virulence factors of E. coli. So we're done of the adhesion, so most of the bacteria will not actually stop there. No, some bacteria are not content just sticking to the surface, so they have the ability to invade the cells that make up the lining of your intestines. So it's just like tiny invaders breaking into a house. So this can actually cause infections and other health issues. So if ever there is actually an invasion of these bacteria, this our body would actually respond to it, no? So it would actually activate our immune response. So aside from that, we have also some toxins production. So we have your exotoxin. So what I want you to remember for exotoxin production is these three toxins. We have the LT, stands for the labile toxins. We have the stable toxins for ST. And we have also the Shiga-like toxins. Why Shiga-like toxin? If you can remember, Escherichia coli um, has the capacity to mimic and to receive virulence factors. That is why Escherichia coli produce a toxin similar to the one made by Shigella bacteria, and that is what we call as the Shiga-like toxin or Shigella-like toxin. So these toxins can be very harmful and are actually produced by certain E. coli strains. And kisama ning mga certain E. coli strains, that is what we call as the Shiga toxin producing E. coli or the STEC. Again, your Shiga-like toxin is produced by your Shiga toxin producing E. coli, Shiga toxin producing E. coli. And then they could actually cause severe food poisoning. So when you say exotoxin, please take note that exotoxin, yeah, it is indeed very common to gram positives. But then again, it is not strictly for gram positive bacteria alone. Exotoxins can be produced by certain gram negative bacteria. And one example we have is your Escherichia coli. Again, when you say exotoxin, think of your LT, ST, and your Shiga-like toxin. Those three, labile toxin, stable toxin, and Shiga-like toxin. And these toxins are usually very common or common culprits for severe food poisoning. So if you could remember, your exotoxin has different subtoxins, and that includes your enterotoxins, right? If you could remember that, when you say enterotoxins, do not be confused. Remember, entero ni enter sa stomach. If it enters the stomach, it usually causes food poisoning. That is the main culprit or the main toxin that would actually cause food poisoning, enterotoxin. And enterotoxin is a subclassification of your exotoxins. So on your subclassification, we have also neurotoxins, which you will learn on your later lecture sessions. So aside from exotoxins, we have also the endotoxins. So if exotoxins is very common to gram positive, but then again, it is also produced by some gram negatives, endotoxins is somehow strictly and very common for negative bacteria. Why negative bacteria? Because your endotoxins are part of the outer membrane of your gram negative bacteria, right? So the most important part of that portion is the lipid A. If you could remember, the lipid A portion of your lipopolysaccharide that is usually seen in your gram negative can help produce and synthesize this type of toxins right here, the endotoxin. Again, your endotoxin is part of the outer membrane of your gram-negative bacteria, and that is your lipopolysaccharide. Again, specifically, your lipid A portion of your lipopolysaccharide helps in the production of your endotoxin, which is common to the gram-negatives. So why is lipid A very important once again? I think I have already discussed this, but then again, for recall. So if when these bacteria when these gram negative bacteria die, they release lipid A, right? So when they release lipid A will not be, most of the time will not be destroyed. So when they release lipid A, this can actually trigger a very strong immune response in our body, which sometimes causing fever and inflammation as well. So we have here your iron binding siderophore. Your iron binding siderophore, on the other hand, this obtains iron from human transferrin or lactoferrin, meaning class bacteria also needs iron to grow. We need iron to grow. And it is very important to remember this one, class, because these iron siderophore or iron binding siderophore has the capacity to steal the iron away from our body. So that is the purpose of your siderophore. Excuse me. Again, just like humans, um, bacteria also needs iron to thrive and to live. And we have these bacteria have this kind of structures, what we call as the iron binding siderophore, which steals away the iron. It steals away the iron from our body. One example of iron binding siderophore is your aerobactin. Aerobactin. So I will show you later on another slide for the next slide. So that is your aerobactin right there. So aerobactin. So bullet number 3. So aerobactin is an example of your siderophore. Okay? So it steals the iron away from our body. So what we have here is the strain for E. coli, and that is what we call as the UPEC state strain. So it stands for Uropathogenic E. coli. That is the UPEC, Uropathogenic E. coli. So E. coli, again, is the most common cause of urinary tract infection. Sorry. So it is the most common cause of UTI, specifically in humans. And why is that so? This is due to its ability to stick to the walls of the urinary tract and evade the body's immune defenses. If you could remember, it has the fibrae and the pili, and with these structures, it actually helps the bacteria stick to the walls. That is why those female patients, usually UTI are very common to female patients, but then again, it is also common to it is also possible to male patients as well. But again, those who suffered from UTI can really tell that during their infection process, they would really go. They have the urge to urinate frequently. So that is our body trying to wash off the bacteria. But then again, um, washing off the bacteria is not effective because of the pili and the fibrae. So again, we have here the pili. Again, the pili. What is the role of the pili of the UTI infection? So it would actually attach to the epithelial cells. So it would be attached to, they help the bacteria attach to the epithelial cells, specifically in the lining of the urinary tract. So this is very important because their adhesion right here helps the bacteria resist being flushed out by the flow of urine. So that is the role of the pili. We have also here the cytolysine. When you say cyto, that means cells. So that means lice or burst. No? So it's killing. So what is the role of cytolysine in UTI? So these toxins right here uh, could actually kill the immune cells. So specifically your neutrophils that would actually try to fight the infection. So when they try to kill the immune cells, they actually inhibit phagocytosis. So what is phagocytosis again? So this is actually the process by which immune cells or your WBCs try to engulf or eat and destroy the bacteria. So if there are no immune cells, then there would be no phagocytosis. So this could actually mean that cytolysine helps the E. coli evade both the immune response, making it harder for the body to clear the infection because there's no immune cells anymore fighting for the infection. And then again, we have here the aerobactin. Aerobactin is a type again of siderophore This usually binds and transports iron. So, what is the role of aerobactin in UTI? So, iron is again essential for bacterial growth and metabolism. So, in here, aerobactin allows E. coli to steal iron from these proteins, from your lactoferrin and your transferrin, if you could remember. So they could actually ensure that the bacteria have enough iron to grow and thrive, even in the iron limited environment of the urinary tract. That is why bacteria can grow, even if there's no iron, even if our urinary tract is limited for iron, because they can actually steal iron from our cells. That is the purpose of your aerobactin. So what we have here, we're done of your extraintestinal pathogens or extraintestinal strains, and that is your UPEC. That is the common cause for your UTI. But for your gastrointestinal pathogens, it is subdivided into five major categories. We have here your ETEC, AEC, EPIC, EHEC, DGEC, and AEC. And we would actually discuss this one by one. But before that, I will be showing you this table. As you can see here, you could appreciate in this table. You don't need to memorize everything, but you could actually appreciate that there are actually relevant serotypes present in each E. coli strain right here. So we have relevant serotypes that you don't need to memorize, but then again, appreciate that these bacteria has the capacity to grow and mutate, no, molecularly. So we have here some serotypes. So let's proceed. So let's start with your enteroadherent E. coli. So your enteroadherent E. coli is actually subdivided into two types. We have the enteroadherent E. coli or enteroaggregative E. coli and your adherent DIEC, and that is your diffusely adherent E. coli. So let's start with your enteroadherent E. coli in general. So adherent, so this is a type of strain of E. coli that would just adhere to the cell lining in the intestines or in the urinary tract. So this is the type of your E. coli that cause infections by sticking or by attaching to the cells lining in the intestines, causing diarrheal syndromes. So this type of diarrhea, this type of diarrhea that is commonly caused by your enteroadherent has no presence of WBCs and RBCs, meaning tama na siya attachment. Tama na siya attachment. So it would stop there. So they would just adhere and there's no invasion. Therefore, there's no presence of WBC and RBC. So again, there are actually two main types of your enteroadherent. And it is actually based on how they adhere to the epithelial cells. So what we have here, number one, is your diffusely adherent E. coli. Your diffusely adherent E. coli. So how does this work? So your diffusely adherent E. coli would adhere to the epithelial cells in a diffuse pattern. So what does it mean? Meaning they spread out evenly over the surface. So that is why they are known as diffusely adherent E. coli. So they adhere to the epithelial cells in a diffuse pattern, meaning they spread out evenly over the surface. So we have certain infections that this strain of E. coli would cause. It could cause UTI, just like your UPEC. It could cause cystitis. What is cystitis? That means bladder infection, which is common to children. We have also here pyelonephritis. What is pyelonephritis? That is kidney infection, which is very common to pregnant women. So aside from that, we have it could actually cause gastrointestinal tract again. So it is one culprit of a diarrheal syndrome with no presence of WBC and RBC. Again, diarrheal syndromes without WBC and RBC. So let's go to your next type. So that is your enteroaggregative E. coli. So If your diffusely adherent E. coli sticks to the epithelial cells in diffuse pattern, which they could actually spread out evenly over the surface. Your AIAC, on the other hand, or your enteroaggregative E. coli, adheres to the cells in the intestine in a unique way. So this unique way is what we call as the stacked brick pattern. So when they observed use under a microscope using the HEP2 cells, so HEP2 cells is a type of human cells used in the laboratory for research. So it is actually derived from the epidermoid carcinoma of the larynx, if you could remember your histology. So this is an example of your stacked brick pattern, which is observed again in your AX. So this is the stack brick pattern. Again, just a review for key points to remember. Again, your diet, diffusely adherent E. coli. So usually these type of strain adhere to the cells in a diffuse pattern. So again, they are actually spread out. They spread out evenly over the surface. We have also here your enteroaggregative E. coli that adheres to the intestines in a unique way described as the stacked brick pattern, which uses this HEP2 cells. This is again a type of human cell used in laboratory for research. Please remember that these two types of diarrhea is a type of diarrhea without the presence of WBC and RBC, adherent diarrhea. So what we have here is another type of diarrhea, and that is your enterotoxigenic E. coli. So what is your enterotoxigenic E. coli stands for, or your ETEC? So this is actually a type of diarrhea that causes traveler's diarrhea. If you could remember, what type of amoeba, or not amoeba, parasite that causes traveler's diarrhea? If you could remember, that's Georgia lamblia. So in bacteriology, the common culprit for traveler's diarrhea is your Escherichia coli ETEC, also known as your Montezuma's revenge or La Turista. So why La Turista or Montezuma's revenge? So the term Montezuma's revenge is uh, common is a nod to the Aztec Emperor Montezuma II. So Montezuma met a very tragic end at the hands of the Spanish explorer that is Hernan Cortes. So it is actually a playful way to refer this type of diarrhea, traveler's diarrhea, suggesting that Montezuma's spirit is actually causing the mischief for modern travelers, especially if you want to travel places like Mexico. So that is why some of the travelers, Western travelers, suffer from diarrhea. And the main culprit there is ETEC, because ETEC is very common and commonly isolated on Mexico. So what type of diarrhea ETEC usually cause? So ETEC likes to stick to the upper part of the small intestine. So upper part of the small intestine. That is why proximal small intestines right here. So this is where it starts to cause problems. So but then again, if you could remember here, the infective dose, you'd need to swallow a lot or consume a lot of ETEC bacteria. So somewhere between 1 million and 2 billion. So if you have consumed this number, then you probably get sick. So this usually happens through contaminated food or water. So how does ETEC usually makes us sick? So ETEC has toxins. So ETEC produces these two main toxins right here that makes us feel sick. So we have the heat label toxins and then the heat stable toxins, if you could remember, right? So this is what type of toxin again? This is a type of an exotoxin. So these two types of toxins can cause watery diarrhea. Both of them causes watery diarrhea. So for your heat label toxins, uh, your heat labile toxins usually gets destroyed by cooking, or it actually get destroyed when it is exposed to heat. But for your heat stable toxin, it is tougher because it can survive heat. That is why it is known as the ST, or the stable toxins. But then again, it has the same manifestations or pathogenesis. Why? Your toxins right here um, usually causes or activates an enzyme called adenylate cyclase right here. So what is this enzyme? So this enzyme usually causes the intestinal cells to release too many electrolytes like your sodium and your potassium. So if they could release too many electrolytes and fluids into the gut, so it's just like where the sodium goes, chloride goes, and then water follows, then it could actually, this excess fluid in the intestines could actually leads to watery diarrhea. So it is also the same pathogenesis of your stable toxins. So your stable toxins, it uses or utilizes this enzyme instead of adenylate cyclase. It uses your guanylate cyclase. So this enzyme also has the same purpose. It usually causes the intestinal cells to secrete excess electrolytes and water into our intestines. So the result is actually the same. So it usually causes watery diarrhea. So what we have here is the enteroinvasive E. coli. What I want you to remember here is the virulence factor. The virulence factor is the invasin, INVASIN, invasin. So, although what we have here, enteroinvasive E. coli can cause Shigella-like infection or dysentery-like infection. Although this, although AIC and Shigella species are morphologically similar and produce clinical disorders, the infective dose in AIC that could produce disease is actually much higher compared than of that the Shigella. So, effective dose of your AX could be 1 million, but for Shigella, about 100 bacterial cells, about 10 to 100 bacterial cells. So, if we could actually get this dose, you could actually be infected with Shigella. So, for AIC right here, for the pathogenesis of AIC, your enteroinvasive E. coli usually enters and attaches to the cells lining in the intestines. But then again, it will not be contented just by attaching. So after entering or after attaching, your AIC usually invades the intestinal cells, moving from one cell to another. That is why it could actually cause destruction. So from direct penetration, it causes invasion and destruction. Again, so your enteroinvasive E. coli right here penetrates, attaches to the cells lining in the intestines. Right after attachment, it would invade the intestinal cells, moving from one cell to the next. As it moves and as it grows, the AIC would actually destroy the intestinal lining, causing bleeding and inflammation. So, in most cases, we could see WBCs and as well RBCs. So, please include there, right? RBCs. So, if the diarrhea has the presence of WBC and RBC, we call that dysentery. Why dysentery like or Shigella like infection? Because the type of diarrhea Shigella causes is a dysentery type of diarrhea. That is why Shigella is the main culprit for bacillary dysentery. That is why this type of or subtype of E. coli is also known as the Shigella-like infection and dysentery-like infection, your AIC. So next, we have here the enteropathogenic E. coli. So the infection usually causes the infantile diarrhea. What I want you to remember here is enteropathogenic. So pathogenic, you could remember pedia. Although infants are not pedia, just think closer to age raman ang pedia. So pedia pathogenic infantile diarrhea. So usually, studies relating to epic strains are some characteristics that are not usually seen in ETEC and usually not seen in AEC, but the your epic strain has the combination of both. So, epic primarily causes your infantile diarrhea, meaning which is very common type of diarrhea in young children. So, as you can see here, a comparison with other E. coli types, unlike ETEC right here, which is toxin mediated, your EPEC uh, do not cause do not produce any toxins. Again, unlike ETEC, which causes diarrhea through toxins, and AEC, which here invades and damages to the intestinal lining, EPEC has a different strategy of operating. So, if ETEC produces toxins, it is mediated by toxins to cause watery diarrhea. And if AEC right here invades and destroys intestinal cells, causing dysentery, EPEC on the other hand, or your enteropathogenic E. coli doesn't produce these toxins, and it also isn't as invasive compared as your AEC. So how EPEC causes disease? So EPEC has the ability to attach to the borders of the intestinal cells. So it actually clings there to the lining of the intestines without deeply penetrating or destroying the cells. So they actually stick well, so they don't tear, or they do not invade deeply like the other strains of other bacteria. So that is why the attachment of your EPEC, or the adhesive property of your EPEC, disrupts the normal intestinal function. So just like other symptoms of diarrhea, this could actually cause watery diarrhea, and the presence of the bacteria could also irritates the intestines. That is why the children or the patients that could suffer from EPEC also causes would also experience watery diarrhea, and could actually also, what is very common to EPEC is the presence of mucus in the stool. So if you could remember, it will just stick to the intestinal cell border. So it disrupts the absorption. It disrupts the intestinal wall. So it could actually produce mucus as well in the stool. But then again, it do not damage the host, or they damage the intestine. So that is your EPEC. So what we have here is your EHEC. So EHEC is the most pathogenic bacteria that is under your E. coli. So EHEC stands for enterohemorrhagic E. coli. It is also known as your virotoxic E. coli, or VTEC, or your STAC. What STAC stands for? Your Shiga toxin producing E. coli. E. coli. So, what we have here are your virotoxins. When you say virotoxins, usually they damage the lining of the blood vessels. Again, your virotoxins right here, the two of them, damage the lining of your blood vessels, particularly in the kidneys. So, they damage the lining of the blood vessels. So, what differs from your virotoxin 1 and virotoxin 2? Your virotoxin 1 and virotoxin 2 are biologically similar. So they are biologically similar but immunologically different, meaning your virotoxin, which can be neutralized by your antibodies to Shiga toxin, your virotoxin 2 cannot be neutralized by these same antibodies. Again, they are biologically similar, but then again, immunologically different. Virotoxin 1 can be neutralized by the antibodies, while virotoxin 2 cannot be neutralized by the same antibodies from your Shiga toxins. So what I want you to remember in your enterohemorrhagic E. coli is it could actually cause severe infection or disease. One infection is what we call as the hemorrhagic colitis or hemorrhagic this one hemorrhagic uremic syndrome. So what does this cause? So your if you could remember again, your EHEC toxins, the main pathophysiology of your EHEC toxins could cause damage to the lining of the blood vessels, particularly in the kidneys. So just think of this, if there's actually damage, so for a while. If there's actually damage, let me explore and maybe I can write something. No, I can't write something. Sorry. So, if there is actually damage, the damage can cause abnormal clotting. So just you could actually draw your blood vessels. So draw your blood vessels. So your virotoxins are like knives that would cause damage of the blood vessels. So maguba imong blood vessels. So what will happen to our body? Our body would actually tell the platelets to fix the damage. So this damage would actually cause abnormal clotting because your platelets would try to fix the damage, no, yung pikitan ang mga buslot nga blood vessels. So if there's abnormal clotting, just think of this as small clots in the tiny vessels. So these are small clots. Your RBC would actually pass through these tiny vessels. If naa gani mga clots, if they're actually minor clots, your RBCs will get destroyed. So your RBCs will get destroyed. Why? As these small clots form, they could actually lead to the mechanical destructions of your RBCs. So just imagine when your RBCs pass through these damaged or obstructed blood vessels, so they could actually be torn apart. So they are actually torn apart, resulting in what we call as the schistocytes. So this is what your schistocytes look like. So magupa ay mo ang RBC. So if this is your normal RBC right here, so if your RBC would pass through these damaged or obstructed blood vessels, magisi siya, no, ma-half siya into two. So that is why we have here the schistocytes. So matunga siya into two. So if naguba ang RBCs, there is actually hemolysis. So the process of breaking down the RBC is known as hemolysis. If you could remember your hematology. So there is hemolysis. So in hemolysis in your hemolytic uremic syndrome, hemolysis is the key feature and could actually contribute to the low levels of the RBCs. That is why people suffering from your enterohemorrhagic disease could also could also presents low levels of RBCs or what we call as anemia. So in your hemorrhagic colitis, naguba na ang imong hemorrhagic colitis and your hemorrhagic uremic syndrome, naguba imong kidneys, naghemolysis pa jud ka. So you could actually experience bloody diarrhea as well. So that is why your HUS. So another again, the indicator for that is the presence of your schistocytes. SC, what is the spelling of schistocytes? And that is your SCHISTOCITES. T TS, schistocytes. So, Bonisha, this is your schistocytes right here. So that is the clinical significance of your E. coli. So the presence of this one, of your schistocytes in the blood, is actually a sign of ongoing hemolysis. And it could actually often help the doctors to diagnose and monitor the HUS or the hemorrhagic uremic syndrome. So again, your enterohemorrhagic E. coli is correlated to your HUS. So how are we going to get this one? That is actually through the ingestion of poorly cooked meats. So if you're fond of eating hamburgers, then there is a very big possibility, if that is raw and not cooked properly, we could actually get enterohemorrhagic E. coli that could actually further and become severe as your HUS. So again, this is your schistocytes observed in a blood smear. So it could actually diagnose in helping diagnosis your HUS. So your HUS class is um, common to a certain serotype. And that certain serotype is what we call as your 0157. So your E. coli strain serotype 0157HC7, also known as the culprit of the Hamburger's disease. So that is the main culprit of your enterohemorrhagic disease or your E. coli serotype 0157H7. Why this is associated with Hamburger's disease and why this is associated with hemorrhagic diarrhea. So your EHEC, which includes the association with hemorrhagic diarrhea and HUS, is also associated with the famous Jack in the Box incident right here. So what happens in your Jack in the Box? So, in your Jack in the Box incident, way back in 1993, there was actually a major outbreak of your E. coli 0157H7. So, there was actually a link to undercooked hamburgers from that fast food chain, Jack in the Box. So, most of the people who went to that fast food chain to buy burgers, they experience severe health problem. So some of them actually died. So some of them are suffering from severe cases. So 178 people suffered from severe cases. So, in here, how are we going to detect this type of strain, this main culprit of hemorrhagic disease or hemorrhagic diarrhea? So, your some diagnostic tests that would identify E. coli includes a mug test. What is a mug test? That stands for four, four as in number four, methyl umbelliferyl, UMBELLIFERYL, beta D-glucuronide. So that is 4-methyl amberoliferyl beta D-glucuronide. You can Google it if you want. So that is the MUG test. So this test is used to detect the presence of certain E. coli strains. So that certain E. coli strains we are talking about is the E. coli serotype 157H7, which causes the enterohemorrhagic E. coli. So your E. coli 0157H7 is smog negative. So it is smog negative. It is smog negative, unlike the common strains of E. coli, which is smog positive. Again, your MOG test differentiates E. coli from the your E. coli 0157H7 from the most common E. coli. So your E. coli 0157H7 shows negative results in your MOG test, while the most commonly isolated E. coli shows a positive result. It is also true to the sorbitol. So please take note that your E. coli strain 0157H7 is sorbitol non-fermenter, meaning it would not ferment sorbitol. It won't and can't ferment sorbitol. So therefore, we would be using a specialized agar, and that is what we call as the sorbitol McConkey agar. So it is a McConkey agar with an added ingredient, and that is the sorbitol. So sorbitol or your SMAC, since your E. coli 0157H7 cannot ferment the sorbitol, When it is inoculated on your sorbitol McConkey agar, it shows colorless colonies. Unlike from your ordinary, commonly isolated E. coli, when they are inoculated in the smack, they shows a lactose or pink colonies. So, in here for the smack for E. coli 0157H7, since it cannot ferment your sorbitol, it shows colorless colonies. It shows colorless colonies because it does not ferment sorbitol. So, how are we going to um, further differentiate this strain? We could still do the stool examination. So patients infected with E. coli 0157, which is very unique, please take this down, typically have stools that do not contain WBCs. So they do not contain WBCs. Again, your enterohemorrhagic E. coli diarrhea that is caused by your E. coli strain serotype 0157H7. When you do stool examination, their stools do not contain WBCs. So meaning the absence of WBCs would actually helps us distinguish EHEC infections from the other types of bacterial infections where WBCs are present. Okay? So this is actually a story of Brian Krinner. So since one of the victims of the Jack in the Box, Brian Krinner just went and asked for a burger from her dad. So it actually starts when she ate hamburger from the Jack in the Box fast food chain. So little did she know that the hamburger was contaminated, particularly from a harmful strain, E. coli 0157H7. So the strain produces a toxin that could actually cause severe illness. And in Brian's case, it actually led to a condition called HUS. So that is what we have discussed a while ago, the hemolytic uremic syndrome. So again, your HUS is a very life threatening complication that could cause kidney failure. And in Brian's case, it caused her to bleed from every orifice and it requires months of dialysis. So, every orifice of her body, sa iyang eyes, sa iyang ears, sa iyang nose, kasi siya bleed. So, it actually required months of dialysis kay bati na kay siya condition. So, the severity of her condition was so extreme that she slipped into a coma. So the doctors had to remove her large intestine and place her on machines to keep her heart, lungs, and kidneys functioning. Although Brian eventually woke up from the coma, her life never was the same. So dili na jud the same. But then again, these actually helps not help Brian, but Brian helps us in being aware of this strain. So Brian was 12 years old of this, if I'm not mistaken. He's like, no, he's 20 years old at this time, but she still needs to practice her speaking. Her cognitive skills are not the same anymore. She was asked last year, "How are you, Brian?" So she just answered, "It's going to get better. It's tough right now, but I know it's going to get better." So Brian is just one of the few victims. One of her friends actually died because of eating the E. coli strain. So that is the sad reality of the conditions of these people. Okay, so that was the story of Brian. So spot check right here. So I have a spot check before we proceed to the next topic. Which of the following is the common cause of infantile diarrhea? Infantile, what's your answer? Your answer should be epic. Why is it epic? If you could remember, ehec is not the answer because it is primarily associated with severe gastrointestinal illness, just like Brienne. So it could actually lead complications such as hemolytic uremic syndrome and could actually cause kidney failure. So again, remember what you I want you to remember in EHEC is the known strain E. coli 0157H7, but this is not the answer. Why ETEC is not the answer? ETEC is not the answer because it is known for causing travelers diarrhea. Travelers diarrhea, though it is also common cause of diarrhea in young children, but then again, it has a different pathogenesis. If you could remember, it produces your heat label toxins and your heat stable enterotoxins that could lead watery diarrhea. So please remember that although it is very common as well to children, this type of diarrhea is acute and self-limiting. So, why is it that AYEC is not the answer? So, AYEC is not the answer. If you could remember, AYEC is a type of strain that has similar syndrome or signs and symptoms to shigellosis. So, this type of diarrhea includes bloody diarrhea, fever, and some abdominal cramps. It is AIAC because it invades and multiplies the intestinal wall. Though it is associated with invasive form of disease, it is rather than the it is not as your infantile diarrhea is not as invasive compared as your AF. So the answer here is epic. If you could remember infantile, think of your pedia. Pedia, infantile or infant's pedia. So epic is actually has a distinct strain that has the mechanism of attaching and could cause diarrhea, particularly problematic in infants. So, what is your answer here? Which of the following is associated with your E. coli strain serotype 0157H7? Your answer should be this should be associated with letter A. EHEC. Typically associated with your E. coli 0157H7 or your hemorrhagic E. coli. Just remember again, your hemorrhagic E. coli produces your Shiga-like toxin, your STEC, and could actually cause what again? Potentially life-threatening complications, including your HUS. So we have your other choices. Just a review again. Bahalag balik-balik. So we have your EPIC. It is associated with infantile diarrhea but not linked to serotype 0157. AYEC, on the other hand, it causes dysentery-like illness but is not associated with 0157H7. Your EdTech is known to cause EdTech cause travelers diarrhea and produce enterotoxins but not linked to 0157H7. So what I have here is an example of your SMAC. So the SMAC, what is SMAC? Again, SMAC stands for sorbitol McConkey agar. So what we have here is E. coli 0157H7 on your negative control, and we have E. coli 0157H7 for the positive control. If you could see again, your non-E. coli 0157H7 ferments the sorbitol. So when it is inoculated in your SMAC, it shows pink colonies. So while your E. coli 0157H7 cannot ferment sorbitol. That is why when it is inoculated in your mac, it shows a colorless colony. So if you could see here, if this is your test medium, we could, as a metag, you could tell that the patient is actually suffering from E. coli 0157H7, man, because we have here identified the colorless colony. So along with your commonly isolated E. coli, naagyoy nasa goal nga 0157 with use of your SMAC. Another one right here is an example. So this is again figure A right here. Your figure A is an example of your commonly isolated E. coli, while your figure B on the other hand is your E. coli strain 0157H7. Again, coli 157H7 cannot ferment sorbitol. That is why when it is inoculated in your sorbitol macaque agar or your SMAC, it shows colorless colonies. So while your E. coli ordinary strain of E. coli or your non-E. coli 0157H7, they typically ferment sorbitol. That is why it produces acids, resulting in pink to red colonies on the SMAC. Okay? So what we have here again is the differentiation of your E. coli or how to identify your E. coli. Number one, aside from gram staining, we do the McConkey. If we review again, your E. coli is a rapid lactose fermenter. So as you could see right here, so this is your McConkey. So we have your lactose positive E. coli right here. So we have pink colonies and these are your lactose negatives. Well, on the other hand, we need again your EMB, your acyl methylene blue. So when your E. coli is inoculated in your EMB, it shows a greenish metallic sheen. So it shows a greenish metallic sheen. So this is how it looks like. So um, we have different INVIC reactions right here. We have the indole, methyl red, Volkswagen and citrate. And I will be discussing this in another lecture video. So I will stop here para dili siya taas kaayo. Alright, so I'll see you in the next video.